Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts. And now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. From Microbe TV, this is Immune, episode number 86, recorded on October 15th, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Welcome back. Great to see everyone.
From Cleveland, Ohio, Steph Langle.
Hi there. Hi, everyone. Last time I saw Vincent, we were both, well, I was in Australia. He was conferencing in, and I've sort of recovered. That was my first international trip. It's hard to come back.
Did you like it?
Oh, loved it. Yeah, it was great. It's
Did you see a kangaroo?
We did. We went to the Steve Irwin Zoo and saw kangaroo.
And a wallaby?
Mm-hmm.
Cool. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. I sadly have not been to Australia, but I am still happy to see everyone.
Yes, very happy to see you too.
You've been to South Africa. That's pretty good.
I have been to South Africa.
Mm-hmm.
So, Steph, you said it was your first international travel?
I'm sorry. I should say that's not true. It was. That's not my brain's working right now. It was not. I have traveled internationally before. I think what I mean is that it was my first very long international flight. The international trips I've been to were Costa Rica, so South America, Brazil, but never that far.
Yep, it's far. By the way, on Twin yesterday, we talked about why travel like that and other things screws you up.
Oh, good. Great.
It's all about
that one.
You change your photo period, right? You have a certain light and dark cycle.
Right.
Yep.
And when you change it, that totally screws everything up.
That makes sense.
And this paper, it was amazing paper. They mapped the region of the brain that gets the signal from the optic nerve and puts it into certain neurons that regulates your sleep-wake cycle.
That is really incredible.
It was incredible because they use knock-ins, knockout, CRISPRs, tracing with viruses. Oh my gosh, the technology is amazing. And they end up identifying this circuit, basically, and they use mice which have a photo period too, right?
Right.
Yes.
Now, the cool thing is, it turns out that blind people still get jet lag.
Wow.
Really?
Because there are other receptors which don't see visible light. There's something else kind of light that also cues you, right? That's very cool.
That is very cool. I'm going to have to make sure I take a read of this paper next time I go to South Africa.
Oh.
So, according to Huberman, right? The Huberman podcast. Do you know this guy?
Yeah, I've heard of him. Yeah.
Neuroscientist at Stanford.
Right? Yep.
He said the best way to counter jet lag is to go outside for sunrise and sunset. Just stand out when the sun rises and sets, and then that does it. If there's some kind of light, some special light. I don't know if he's right. Anyway,
helps.
it's very cool. I love when you do science that impacts your everyday life.
Yeah.
Right?
Yeah.
Anyway, but
or explains things that you're curious about because you're like, well, I wonder why.
Yeah.
Yeah.
Well, I think that's the best. And today we're going to do that.
Yep.
So if you like this kind of thing, if you like these science programs, we'd love to have your support. Go to microbe.tv/contribute. So we're going to start with a snippet from Steph.
Thank you. Yes. And I was thinking I don't know if I've done a snippet. I'm like, hopefully I can keep it short. You know me, I tend to go on. So you cut me off if it's just too much. So the paper is entitled Distinct Olfactory Mucosal Macrophage Populations Mediate Neuronal Maintenance and Pathogen Defense. This is in the Society Journal Mucosal Immunology. And the first author is Sebastian Welford. Last author is E. Ashley Mosman. They are at Duke University, and they have published a series of papers over the last couple of years defining the immune response in the olfactory mucosa in relation to viral infection and vaccine responses. But this paper today is really defining specific macrophage populations. And so I'll get into that. But first, the olfactory mucosa, this, if you are listening in sequence, we've been talking about the olfactory mucosa, or really the upper respiratory mucosal space as it relates to viral infections. And so this is very related. This olfactory mucosa is a specialized area of the nasal cavity. And in the paper, they show you very nicely where that is. It's involved in the detection of odors. That's where most people would think of it, because there are olfactory receptor neurons, which are the nerve cells that have the receptors that are designed to detect odor molecules in the air. And once the molecules bind to the receptors, they stimulate the neuron and send signals up to the olfactory bulb in the brain, which processes it and identifies it as a scent. And so it also, though, this mucosal barrier is a mucosal barrier from the upper airways to the brain. So very important. And all of this is interesting immunologically because this is a site of pathogen replication and inflammation. And so they call them olfactotropic pathogens. I thought that was a cool word. Most relevant, we're going to talk about SARS-CoV-2, but these pathogens, viruses, they can exploit this pathway to infect the brain by bypassing the barriers to the central nervous system.
This really surprised me. I guess I hadn't thought about it in that way, but these nerves send out dendrites and they're floating around in the air. Like everything we breathe in is basically has a direct conduit to the brain, which is a little scary. Mm-hmm. And I kind of conceptually knew that, but it didn't really register how vulnerable that area is.
Yeah.
To infection.
The same. There are other things that I remember in grad school, we had talked about some of these cells for a completely different reason. And the fact that they had this vulnerability did not ever come up. And I'm sort of like, hey guys, that might have been kind of important. Maybe we should have talked about that too.
Despite that, though, it's pretty well protected. Not much gets in that way.
That's
right?
amazing. The nuance required is pretty remarkable. And I think what I didn't realize, which by logic makes sense because I continuously smell things, even though maybe things get damaged or I get a viral infection, that these olfactory, I say ola, it's olfactory sensory neurons, they can be replaced throughout your life, especially following inflammation. And they regenerate. They have this regenerative property from local stem cell progenitors. And so you can preserve your lifelong ability to smell things because they do this. This is unlike other neurons that don't do that. And so the immune cells within the olfactory mucosa, they have to perform two very important tasks. They have to support this fundamental process of being able to smell things, but they also have to prevent pathogens from hijacking the olfactory system to access the brain, disseminate within the respiratory tract, or just directly damage the tissue. And so this group is interested in really defining that. And this paper presents distinct macrophage populations within the olfactory mucosa, and what things are they expressing on their surface? What do they look like? Where are they? And what do they do? So briefly, macrophages, we've talked about them before. They contribute to inflammation. They secrete cytokines and chemokines. They present antigens. They activate lymphocytes, and they're really effective at clearing pathogens. They really can. So they clear pathogens by this other kind of side of them, which is anti-inflammatory. So they can do the pro-inflammatory side, but to clear things, you have to phagocytize dead cells, debris, and orchestrate tissue regeneration, which is going to promote wound healing. So two very different things when you're thinking about immunology or immune responses. And macrophages can be very specific. They are very specific to tissues that they're found in. I think maybe the past decade, that's one of the biggest things I've seen and I've learned as a non-macrophage biologist is just how diverse they are. There's this seemingly proliferation of literature showing how diverse they are in different tissues. Microglia are the resident macrophages in the central nervous system. There's been others described at brain borders. There's peripheral nerve macrophages. And there have been macrophages identified in the olfactory mucosa. They just really haven't been well studied. So what this group did was they had mice and they were using single cell sequencing to pull out CD45 positive immune cells. So this is a pan or a general marker on all immune cells. They looked at cells with uninfected and VSV infected mice, vesicular stomatitis virus infected mice, to look at these different populations. Now, they had an issue because they wanted to make sure that the population they were looking at was specific to the mucosa, because when you pull these tissues out, you're going to have possibly contributions from blood coming in because it's highly vascularized. And so they were able to take bone marrow and compare the clusters between bone marrow and the olfactory mucosa to use their single cell sequencing analyses to separate out these clusters. And the clusters of single cells then allow you to dive deeper and look at the gene sets that are associated with those.
So, Steph, you mentioned they collected blood so that they could eliminate those cells. And I think they also, because of where the olfactory mucosa is, when they collect it, they crush up.
They do.
Right. So there is actually bone marrow there too.
Contamination.
So that's why they then also did that bone marrow. And I don't know enough about how you do all that clustering and subtraction analysis and things, but I would think that that is a pretty important aspect of how they got to some of the data. And I guess I just kind of trust them on it. But if somebody is an aficionado in how you do that and whether they use the correct algorithms and all of that, right. I think it's going to really impact how they interpret the data that they have.
Right. Yeah, thank you for that. That's a good point. So, right, why they collected bone marrow was because not only blood contamination, but bone marrow contamination. And maybe that's why previous groups hadn't honed in on these.
Yeah, it's really hard to get them.
Sure.
Yeah. I wondered what Vincent thought about VSV as their model neurotropic virus.
Yeah.
Well, it's not great because it's not a pathogen of mouse, right? But what are you going to use instead?
There are aren't there murine coronaviruses that go neurotropic?
Yeah, mouse hepatitis virus. Susan Weiss has worked on that for many years. Yeah. And also, what's the other one? There's one highly neurotropic rodent virus too. Yeah, I mean, I don't think VSV is great, but a lot of reagents, and that's why people often use it, right?
Mm-hmm. Yeah, right.
But you have to wonder, right? What does it mean? Because it's a totally I mean, now we're understanding that if you have a weird host pathogen combination, you get weird results, right? It may not be normal.
Yeah, so maybe a future work trying to understand are these responses, are they specific to this particular virus or not? But what they found after analyzing the immune cells through their single cell analysis was they had macrophages, dendritic cells, gamma delta T cells, but the two macrophage subtypes that really came out was P2RY12 or purinergic receptor P2Y12. It's a G protein coupled receptor, and it is known, it was discovered on platelets, but it's also found highly expressed on microglia and seemingly these olfactory mucosal macrophages. So that's the first. The second is MHC2 or major histocompatibility complex class 2 expressing macrophages. And these were highly expressed on them. So when you read the paper and you see HI, that's how we define that you can have varying levels of expression of these different markers. And so they are highly expressed on these two populations. And MHC2, would be for those who are curious. This is a molecule expressed on the surface of immune cells that are really important in presenting extracellular antigen fragments for T cells. So CD4 T cell help in initiating immune responses.
If you'd like to know more about that, you can go to my lecture from YouTube from yesterday.
Yeah.
Yes. Okay. Maybe we'll link that in the show notes. Yep. That's great. So they saw after VSV infection, they saw more MHC class 2 macrophages, which is notable. And we'll talk more about that. And just for information, reduced gamma delta T cells, increased CD8s, increased IgA plasma cells, which are in that space quite a bit. But the two macrophage subsets really differed in tissue residency, so where they were located, and that was relevant to what they think the function of these are. So the P2RY12 high macrophages had strong tissue residency, and that was even after the viral infection, where MHC class 2 high macrophages were turning over more rapidly. And so that suggested to them that they were more regularly replenished by circulating monocytes, and monocytes are the cell type that are going to replenish tissue macrophages. And so given this, the differences in where they were seeing them in the mice, the models that they were using, they wanted to see like sub-tissue anatomical niches. And this was helpful for someone like me who doesn't know much about the olfactory mucosa. They describe there's there's layers like an onion, right? The top one is avascular neuroepithelium, and then you have lamina propria and basal lamina, which are really just beneath this neural epithelium space. I'm not versed in these niches and their particular functions, so somebody can chime in and email.
I think those top ones are where the neurons are. There's a lot of neurons, and where they're making those projections out
like
of
the
epithelium, right? So
yeah.
I think that's where the ones that are probably going to be more protective for the neurons are going to be located.
Yep. Mm-hmm.
Yeah.
You mean the macrophages themselves?
Yeah. The P2Y12.
12 high.
Yeah.
Right.
Those guys.
Yes.
P2RY.
And they found over time that the MHC class 2 increased across all the regions, and they thought this was due to maybe environmental exposure. There's more antigens to present, and that the lamina propria probably just undergoes more frequent replacement. But the P2RY12s maybe are longer-term stable residents, to your point, Cindy, that they may be needed to stick around to protect, to interact with the surface, and be responsive to immune responsive. So they did also look at the unique morphologies of the different cell types, which I won't go into too much, but I did want to mention about their function. So the P2RY12, which were associated with maintenance and phagocytosis, they did find that they were more effectively able to engulf olfactory sensory neuron fragments than the MHC2 class 2 high macrophages, which were more linked or more shown to, in their paper, secrete cytokines. And so they thought this enhanced phagocytic ability of the P2RY12, they even showed that this was the case after influenza B virus infection. So they saw that increase more in that subset after a different virus. So they did use a different virus here. Whereas the MHC class 2 were more responsive to secreting something like CXCL10, which is an inflammatory chemokine. So I think they were suggesting that this really distinguishes them as primary immune responders to secrete cytokines compared to maybe more of a neuron supportive role of the P2RY12 that exists above, at the top in that avascular space.
You could sort of imagine that making sense with P2RY being sort of thinking about neuron supportive roles, since it's a receptor for ADP.
Right.
And so it could be kind of monitor, that's the word I want, monitoring the metabolic state,
right.
At least partially of those cells.
Yeah, totally agree. And so that was all found within the context of their mouse models. And so what they wanted to end the paper with was to bring you into some human relevance to make sure that can we find these P2RY12 or something like it, macrophages in human tissue. So what they did was to look at this. They looked at SARS-CoV-2 patients. And why would they look at SARS-CoV-2 patients? Well, because what has been demonstrated that the symptom that you get, which is a loss of taste or smell, is because the virus can infect, and I had forgotten this word from way back when the pandemic started, but sustentacular cells.
Sustentacular.
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 Oh, it's tacular?
Yeah.
Ah, okay.
But they express ACE2, and they are being infected by SARS-CoV-2, which is causing the loss of taste and smell. So,
Steph, I'm glad to know that you also learned that word because of the pandemic.
Yeah, I did not know that word before the pandemic, and I don't think I had read it since then.
In fact, early on in 2020, we had a guy on, Twin, who talked about sustentacular cells and the fact that that's what is infected and causing the loss of taste and smell. Yeah.
Yeah. So from the pandemic, right, emerged a lot of new information, I think, or at least information that wasn't really thoroughly investigated, and those cells were one of them. And so what this group did was they didn't have tissues themselves. They actually reanalyzed previously published.
Can I ask you one question though? Aren't there a subset of patients that don't actually get their taste and smell back, or it takes a really long time?
Yeah. There's some have not, since the beginning, right?
And I think they have that as one of their as one of the subsets are patients with hyposmia long term, after SARS-CoV-2. So yes, I'm sure that speaks to maybe the diversity of our ability to regenerate because they are a regenerative cell type.
So maybe part of what they were trying to figure out was, are these different populations the same or different, right? Because they have two different roles. Of
right. And if they could see in these different patients, and so what they were looking at is patients that did not have this loss of taste or smell, those that did, or those that did long term after SARS-CoV-2.
Okay. Yeah.
So they wanted to, exactly to your point, Cindy, compare, but they focused on. They did focus on both. So the hard thing when you're trying to understand these populations in humans is you're then just trying to match gene sets to the single cell data that this other group analyzed, because you're not analyzing it yourself. So you can't actively in the same analysis compare human and mouse. And so they found that the cluster 4 human macrophages were associated with neuron interactions, phagocytosis, and tissue regeneration, had similar gene sets. So they felt that this was their P2R12-like cluster. And they found that in individuals with normal smell, that they were prevalent, but in people who had reduced smell or smell loss, they were reduced. And so that was suggestive to them that if these macrophages existed in humans and have the role that they found, that they were changed after infection with SARS-CoV-2. So could this represent like this imbalance of inflammatory and regenerative macrophages during SARS-CoV-2? And lastly, to talk about the MHC class 2 high, they found that they had individuals put with COVID shared gene sets more with the MHC class 2 macrophages, whereas people who did not more aligned with the P2RY12. So again, if we're very broadly lumping them based on this paper, so I think we're clarifying that it needs more study. But if P2RY12 macrophages are more regenerative, and those were more likely in the non-COVID patients, maybe that's a response that what they're seeing is these individuals who have this effect can't regenerate their macrophages, and maybe that's why they have loss of taste and smell. So overall, this group was able to identify in mice 2 specific macrophage populations with 2 different roles. One may be more regenerative and regulatory, and one may be more responsive. One may be being replaced by systemic monocytes more than the tissue resident P2RY12. And that's the paper.
Cool.
Yeah.
You just need to look and you'll find.
Yeah.
Right.
Yeah, it definitely gives us lots of kind of interesting ideas and derives lots of more new hypotheses.
Yes.
I thought one of the interesting things that they mentioned in the discussion was allergens, because that's another thing that, we breathe in and probably is going to access that site. And I wonder whether either of these populations are involved in antigen presentation during allergen development, and maybe one or the other promotes development more, and maybe there's a difference in individuals who are more likely to develop allergies than ones that aren't. That was interesting.
You could also wonder, whether they may influence more Th1, Th2 responses.
Yeah, exactly. Yep.
Thanks, Steph.
Yep, sure thing.
Okay. Now we're continuing with SARS-CoV-2, but in a different compartment.
We can't get away from SARS-CoV-2, right?
It does allow for a lot of interesting probing of the immune system, right?
There's
Because
so much
you have a
immunology to
before and
learn.
after.
Yeah, exactly.
Yeah.
So this is a Nature Medicine paper. SARS-CoV-2 specific plasma cells are not durably established in the bone marrow long-lived compartment after mRNA vaccination. It's from a group at Emory University. Nguyen, Hentenar, Morrison, Porter, Solano, Haddad, Castrillon, Rundstrom, Lamoth, Andrews, Roberts, Loniel, Sands, and Lee. So here the issue is, what's going on with SARS-CoV-2 vaccines? Antibody levels decline very quickly after vaccination and even infection, within a couple of months. And even though decline is an important part of antibody homeostasis, it gets slower than it probably should be. So in this paper, they got a brave bunch of volunteers to undergo bone marrow aspiration. Oh my gosh.
Wow. Commitment. Or a lot of money. I don't know how much they offered them.
Some of them serially, some multiple, and then you put a needle in your hip and that hurts. Not that I've ever done it, but I've heard. And then they can say, okay, what's going on in the bone marrow? And so I learned a lot about the bone marrow. When you make a response in lymph nodes to some antigen, you generate antigen-specific B cells, and they become antibody secreting cells. And these give rise to antibodies in the serum or wherever. It could be mucosal tissues, depending on where they are. And most of them die, but some of them make their way into the bone marrow, where they may become long-lived and survive for decades. There's a specific environment in the bone marrow. They have stromal cells and myeloid cells, and they provide all these factors that are needed for survival of these antibody secreting cells. And they become long-lived for decades.
Yes. So there is debate, and I know this more on the T cell side, but it's the same debate in the B cell side about whether there are also some long-lived memory cells that may be generated separately from this. And so whether all memory cells are descendants of sort of plasma cells and antibody secreting cells, or whether some of the cells, when they get activated, directly become memory cells, is something that people like to debate about both types of lymphocytes. And so that may come back at the end here.
So they are going to use as a comparator tetanus vaccination with tetanus toxoid, which antibodies have a half-life for 10 years. You go to see your doctor. Oh, it's been 10 years since you had a tetanus shot. Let me give you another one. So that's what that's based on. Now, influenza, the vaccine only lasts 4 to 6 months in terms of antibody levels. But natural infection, well, if you were infected in 1918, your immunity lasted since then, 90 years. So there's some people who survived that. And then, when H1N1 popped back up in 2009, they had antibodies from 1918. Right. So natural infection apparently is more durable in terms of antibodies.
Yeah, and these 3 vaccines that they're using are 3 different vaccine types. So there's an mRNA vaccine, an inactivated vaccine, and a toxoid vaccine.
Right.
Yep.
So the purpose of this study was they got 19 healthy adults to get bone marrow aspirates. And these were taken 2.5 to 33 months after getting their first mRNA vaccine. All these people got 2 to 5 vaccine doses, and one person gave 3 longitudinal samples over 21 months. A second gave 2 aspirates. I don't know how you would go back after the first one, unless they paid you a lot of money, right?
Yes.
And then some of them got infected also. 5 people reported infection before the bone marrow collection. I'm sure, as you'll see, some of them probably got infected and didn't know it. There's some evidence for that. They all have gotten quadrivalent flu vaccine within 1 to 12 months of the bone marrow aspirate, and they all had childhood tetanus toxoid vaccine. So they used flow sorting to separate cells in this bone marrow aspirate. So you can imagine you don't get a lot of cells from it. So they have to be getting a certain, I think, 3,000 cells per analysis, 3,000 sorted cells, and some of them they couldn't use. So it's a small sample number, and they keep admitting that to their credit. They have in parentheses, small sample number. And use markers to distinguish different populations, right? POPs. They're like POP A, POP B, and POP B, which are in the bone marrow and are making antibodies. And as far as I can tell, it's the POP D that they feel are the really long-lived ones. So POP A and B can make antibody. They have a memory phenotype, but they don't last a long time, right? And so they use the HA of influenza virus for their ELISA assays. They use tetanus toxoid and the proline stabilized spike protein of SARS-CoV-2 to do their ELISAs, right? Okay, so they do these ELISA assays with all of these cells. So they culture the bone marrow cells and they do some sorting into these populations, sort and then culture, and then they do ELISAs. And so all these samples had IgG, all right, just generic IgG, not specific. POP D had the highest amount of flu and tetanus IgG, and they could rarely find SARS spike antibody secreting cells in POP D. So they're looking for antibody secreting cells in these 3 different populations. But they could find antibodies to SARS spike in POP B and A. That frequency is comparable to flu and to HA, but never in POP D, population D. So as Brianne said, it depends whether you think that's all that matters or not, right?
Yeah.
Right.
So I think that we could definitely take that. It seems like the mRNA vaccine does not induce this particular population of B cells.
Yeah.
The CD19 low, CD38 high, CD138 high, which they call long-lived plasma cells.
Yeah.
And so, they don't look, and they look here at those versus a couple of other antibody secreting cell types, but they don't look at the other 2 or other potential long-lived not plasma cells.
They also look at IgA secretion from these populations. Basically the same thing. They find the same distributions among these 3 populations. They find everybody in POP A and B and only the tetanus and flu in POP D. So they conclude that SARS-CoV-2 spike antibodies, antibody secreting cells are largely excluded from POP D.
Yeah. It's interesting. I think that it's very clear the differences.
Mm-hmm.
There's a lot of questions about So CD19 is downregulated on plasma cells. And so that's one of the distinctions, one of the main markers they're using here. And it's clear that there are CD19 positive cells in after SARS-CoV-2 mRNA vaccine that are long-lived. So it's one of those things of, do they have a long-lived plasma cell function? I guess we don't know enough about CD19 positive long-lived plasma cells, maybe to answer that. But it definitely suggests that for whatever reason, and I think we'll discuss, but we need to improve on the mRNA vaccines or another modality if the goal, and that's where we come back to what is the goal? Is the goal to induce a lot of antibodies in a short amount of time like we needed to when the pandemic was emerging and nobody had immunity?
Yep.
Well, now we've kind of shifted where everyone has immunity. And so now it's a matter of targeting vulnerable populations. Are we trying to induce more long-lived immunity so we don't have to get vaccinated as much? So that's kind of like my one general comment. The second one is people get, did they have background on were these people getting vaccinated? How many flu vaccines have they had? They can get flu infected. So the timeline of like the induction of tetanus and influenza specific responses aren't clear. I don't think we have that data.
Let's see if they don't. I didn't see the information on what flu vaccines. They just say they got quadrivalent, within so many months.
Okay.
Yeah,
but
they don't have that information.
These are adults, so I'm sure they've gotten multiple. Right?
Yeah.
Over their lifetimes. Yeah.
So that's kind of, you can't control for that. I would have loved to kind of look at one of these classic vaccines like yellow fever vaccine that is thought of to induce kind of the longest immune response, the longest long-lived plasma cell response. As a comparison, that would have been neat.
Or polio, because everybody gets polio vaccine.
Right. And probably everyone doesn't get yellow fever unless you travel.
If you travel.
Right.
But everybody in the U.S. at least, which is what this population is from, gets polio vaccine. So that's relatively long-lived. At least we think so. Maybe we would find that it's not. I don't know.
But the one nice thing about the yellow fever vaccine, however, is if we had this population getting a yellow fever vaccine, because not everyone gets it, we'd probably have a lot of data on when they got it and number of doses.
True.
Right.
Yep.
Yeah, true.
Which I just help you to normalize like what we're comparing.
Yeah.
Because we don't know if these plasma cells have been induced recently. I mean, they're long-lived in terms of the phenotype, but we don't know when that stimulus was.
Right.
So, a caveat.
I don't know. I think the
ahead.
goal would be to not have to vaccinate so often, right, Steph?
Yeah.
Correct. You're right. I think the overall, what we're coming away from this paper is there's clearly a difference, and we need to understand why, which would probably make it better.
Yeah, I mean, I 100% come away from this paper with there is definitely a difference.
Yeah.
Yes.
But what I don't come away from this paper with is that difference good or bad?
Yeah, and is it like, is this what we're targeting then? Is it like this population that that we want to improve upon? So
I was a little concerned when I read the title that, are people going to think that the mRNA vaccines are not worthwhile because they're not inducing?
Yes, I have seen this paper taken that way.
Right?
You go to the highly critical, formerly known as Twitter platform, yes.
But they have infection in this study as well, and they also did not induce it. So it seems something to the virus and its proteins rather than the vaccine formulation.
Well, I think this is where we could maybe talk more about how we're defining long-lived bone plasma cells in the bone marrow, because as I look at a 2021 paper by Ali Elabedi's group, the title is SARS-CoV-2 infection induces long-lived bone marrow plasma cells in humans. So I think piecing out the methods to try to understand why are they finding completely different results.
Well, one of the counter arguments that people made then, and you said that paper was when, 2021?
2021, right.
So what we were doing was saying we think these are long-lived plasma cells based on the way we identify and characterize what we think are long-lived plasma cells to other infectious agents, right? Because for SARS-CoV-2, it wasn't around long enough to know truly whether these were long-term, right? That's one of the criticisms still of this paper is even a few years out, and they're doing these bone marrow aspirates and looking at these cells, is that still only a few years, where when you get your first tetanus vaccine, you're in childhood. So all the adults, it's going to be, 20 plus or way more years since you had that originally. Even flu, right? Most of the flu vaccines you're going to get for the first time will be earlier in childhood, as early as 6 months, right?
That's a good point. So it's like, do we just need to wait 10 more years to really know what a long-lived plasma cell looks like?
Right. Like maybe we haven't had enough time for those long-lived cells to emerge.
I see what you're saying.
And they might, once they establish, they may proliferate at a low level, and over time that percentage will go up from the 0.1 or whatever percent they have now to the 3 or 4% versus the other two populations, because I don't know though, because the exposures may still continue to happen, right? So you may still have more sitting in population A and population B, and you may never solidify that population D. Maybe there has to be a lack of exposure to the antigen for a certain amount of time in order to establish that niche, right? Because one of the things they say is, when you get the vaccine for tetanus, hopefully you're not actually getting exposed to tetanus during that period of time, right? But for the flu, they say most people will get their first infection by the year 5 of life, and that you get it about every 3 to 7 years. So that's a pretty long time between infections, generally. So maybe it gives that time for that long-lived plasma cell, population D, whatever they're calling, to rebalance and be relatively higher in percentage relative to the other two, because there hasn't been as recent exposures. But when we're talking about SARS-CoV-2, most people have been affected multiple times over the last few years. So there's very little rest time in order for those populations to come out of A and B and establish D.
And that goes back to the comments that you made earlier about having data on vaccination and things like that, because, I get a flu vaccine every year. So you point out something about, it's 3 to 7 years between exposures, but in theory, I have some B cells that are responding to flu yearly.
Yes.
And so would that look different than someone who had that longer period of time between exposures?
Yeah.
All right. So there's a little more data here, then we can speculating. So these results they've obtained are from ELISA, actually ELISpot assays, where the cells are right on the plate and they look at reactivity right there. So they say, let's put these bone marrow cells in culture, take the supernatant and measure antibodies just to validate. So they do it for all 3 populations, pop A, B, and D, and they culture them. They take the supernatant, and then they do a binding assay for the antigens, the tetanus, flu, and SARS-CoV-2 spike. The results are the same, basically. The antigen specificities from the ELISpot are validated in cell culture. Again, exclusion of SARS-CoV-2 ASCs in this POP-D. They also ask whether there's a correlation between the time of vaccination and the bone marrow aspirate, right? That was different among all the subjects. Doesn't matter. Little bit of time, a lot of time doesn't make any difference in the POP-D spike reactivity, SARS-CoV-2 spike reactivity.
I'm trying to remember what their definition of a lot of time was.
I think it's between a month and...
like a couple years, right?
Okay.
Let's see. We can go back to the patient description here. Okay. So the 2.5 to 33 months after receiving the first dose of vaccine, that's when they did the aspirates. So it varied a long time, right? But that doesn't make any difference. You get the same results. What about infection? They have some patients who were also, they're vaccinated and infected. They have 5 of those. They have people who are vaccinated with no self-reported infections, 14. And they found no differences between those 2 groups, right? So whether you're infected or not, you still don't get these long-lived plasma cells in the bone marrow specific for spike. Okay.
But I guess, is that because we haven't waited long enough? We don't have an influenza infection comparison, right? To like,
I don't think they have that.
They didn't have documented influenza infection. So maybe after influenza infection, you also don't see a boost in the long-lived plasma cells because it takes more time, to our earlier point. So I don't know if that means that SARS-CoV-2 infection doesn't induce long-lived plasma cells. I think we're just repeating what we said before. Maybe it's a timing thing.
Yeah.
We need the immunologist of 2080 to tell us the answer.
Right.
Yes. Can you imagine how old we'll be? I don't even. Yeah.
So then they did a kinetic study up to 38 months after the first vaccine. Wait a minute. Hold on. Yeah. Up to 38 months after the first vaccine. And so they're looking at serum antibodies now, right, in this experiment, not bone marrow cultures, to see how that correlates with the bone marrow results. So they see a decline of spike antibodies, spike IgG in serum 3 to 6 months after the first SARS-CoV-2 vaccine. One patient had a booster 7 months after the first vaccine, which showed a rise and fall in antibody titers, as you would expect. And during that time, the flu IgG and tetanus IgG were constant, right? Because that's a nice control for that. And then the serum antibodies go down unless you're boosted again. So, I don't know. I guess it's kind of paralleling what's happening in the bone marrow. Okay, what's the relationship between these systemic antibodies and bone marrow? They looked at titers in the serum, which we've just talked about, and the frequencies of bone marrow IgG antigen secreting cells, and that includes the long-lived and the non-long-lived. And they found for SARS spike a correlation between serum IgG and bone marrow IgG from non-long-lived plasma cells or total antibody secreting cells, but no correlation with the long-lived plasma cells and what's in the serum, right? And that's not the case for flu or tetanus antigen.
Well, flu is kind of in the middle, right? There's like modest correlations with each, but the tetanus is really interesting. And, it comes back to your point, Brianne, that the different types of vaccines, and somehow the toxoid just like nails them right in that long-lived plasma population.
Right.
And those are the main ones making the antibody. Because there's a very tight correlation between the long-lived, what they're calling the long-lived plasma cells, and the serum antibody levels for the tetanus antigen, which is different from flu. It's kind of wimpy in the middle. It seems like the serum antibody is made by both what they're calling long-lived and non-long-lived.
Right.
But then with the SARS-CoV-2, it's all coming from these non-long-lived ones.
Right.
And then the next to the last experiment, they do kinetic analysis where they had these serial samples from 2 patients. So one gave them 3 bone marrow aspirates over 23 months. So those were taken 2.5, 14, and 23 months after the first vaccine. And they do their analysis of the 3 populations, and they see an increase in the frequencies of spike IgG antibody secreting cells in POP A and B at 14 months and 23 months compared to the first. Those were when the boost happened, right? But in POP D, nothing in the first 2 and 0.3% in the last one. So again, kind of mirroring what we've seen before. And the antibody secreting cells are always higher in POP A and POP B. They found the highest flu and tetanus frequencies in POP D, which is what they're saying is the important one, followed by POP B and then POP A. And those flu and tet frequencies were consistent over the course of 2 years in this patient. In the serum, the flu and tet IgG were stable for this period. That's pretty cool, right?
Yeah.
Then for SARS-CoV-2 antibody, there's initial decline at 3 to 6 months, and they remain low for about 7 months. And then they went up, which corresponds to the increase after the boosts, right? Oh, sorry. So they went up at, no, that's not right.
It went up, but they didn't have a boost, but they are suspecting it was an asymptomatic infection, right? Because the antibodies went, but they didn't.
Yeah, this increase occurred without any additional vaccination. Yeah.
Yeah.
And stayed elevated for about 6 months,
And then,
which they say probably there was an infection.
yeah.
They also have a second subject where they got 2 aspirates at 28 and 33 months after the first vaccine. Similar findings, as we've already said for the first one. So,
what wasn't clear to me is whether either of these two individuals had a flu booster during that time frame, because that's really important.
mm-hmm.
Because if those plasma cells in the bone marrow in these different populations stay steady, even with a booster, that's very different from what we're seeing with SARS-CoV-2, right?
Yeah.
Right.
And then the last experiment, they calculate the number of individuals with spike positive responses for each bone marrow ASC subset. And so Pop A, 6 out of 8 individuals, 75%. Pop B, well, everybody, 15 out of 15 had antibodies. But in Pop D, 35%, 6 out of 17 had antibody to spike in the Pop D population, and very low, as we've seen before. So they conclude that the short-lived antibody responses in the serum to SARS-CoV-2 mRNA vaccines may be explained by exclusion of the spike-specific antibody secreting cells from this compartment. There's a nice figure that shows that, we have the generation of antibodies in the lymph nodes, and then we have it for flu, tetanus, and SARS-CoV-2, and then the antibody secreting cells in the circulation, and then they're getting into the bone marrow, and tetanus and flu, they make long-lived plasma cells, but not SARS-CoV-2. I mean, it's black and white. It's too black and white. It's 0. Right?
Well, I think this can be a niche issue, right? Because then you wouldn't generate this population D to any antigens. So it has to be something, either what was happening during the time in which those antibody secreting cell precursors to the long-lived plasma cell were being made, and some kind of cues that they got then that just prevents them from getting to that final destination. Or they will get there eventually, or it's just, a population effect, right? Because if there's more in A and B, there's less in D percentage-wise. And if over time A and D go down, then percentage-wise D will go up, which is coming back to that, exposure and length of rest time in between and things. Yeah. I don't know.
Yeah. And so it's hard to say, is this vaccine modality, which I think is what they want us to say, or is it the timing?
But I feel like I read in here somewhere they mentioned something about the adenovirus vaccine had similar effects.
So they say advectors. They reference 2 other papers.
Yeah.
It's the antigen.
Well, yeah, I mean.
Yeah, it's the same antigen. Right? Yeah.
It's the same antigen. I remember that I teach undergrads and I'm oversimplifying something here. But, when I think of, in particular, an inactivated vaccine or a toxoid vaccine, I think about vaccines that are predominantly, pushing towards antibody responses.
Yep.
And we're not seeing the same kind of T cell responses. And both adenovirus, actual infection, and mRNA, we think of as being sort of a more T cell biased vaccine. And so I think it could still be, the fact that infection, adenovirus, and mRNA are coming out one way, and toxoid and inactivated are coming out another way, I think still could be a vaccine modality thing and not just an antigen thing. And you'd really need to do a head-to-head of the same antigen in different modalities, which you can't do in human.
And stepping back one more point, which is better?
Right, exactly.
Right, because we don't really know the correlates of protection. Everybody's been arguing that, the mRNAs must be better because they generate a T cell response. And so if they don't generate these long-lived plasma cells, is that a bad thing?
Right.
Or are we better off because they generate T cells?
Right. Maybe it's okay. Right. Maybe for long-lived protection and otherwise, healthy population, because I think we could argue that in vulnerable populations, there's a lot of other issues.
Yep.
Their T cells are decreasing, which means if it is T cell bias, then they're more susceptible. But maybe we don't, we care as immunologists, but maybe we don't care in terms of vaccine development. Although I think the point, it's fair to want a better vaccine that lasts longer. So I think,
as long as antibodies are the thing that you need.
Right. Like maybe you care based on the pathogen. Maybe for some pathogens, you really want those antibodies. And for other pathogens, you really want the T cells.
Well, I think when I think about public health perspectives, when we have these surges of people dying, they're elderly people, but they're getting the virus from healthy people who are transmitting. And if antibodies are the correlative protection against transmission, which animal models and some humans are suggesting, then we really do need to care about boosting short-term antibodies so that we can protect the vulnerable.
Yeah,
That's fair.
The trade-off is, though, that you get vaccine fatigue, right?
Yeah.
For sure. That's in an ideal world, right?
And then that affects other vaccines, which is a real problem, right?
Yeah. And will be a real problem if, for example, if we have like an H5N1 pandemic. I'm not saying that's happening, but we would not be ready to do mass H5 vaccinations because of the fatigue of SARS-CoV-2. So
one thing that would have been interesting would if they had a cohort that got Novavax protein vaccine.
Yeah.
Right.
Yeah.
So it's not vectored protein, but it's. Frankly, I think it wouldn't matter, but
because you think it's an antigen issue.
Well, they have a discussion here about it being an antigen issue, which it's a little. They say basically coronaviruses lack highly repetitive organized structures, and most RNA viruses that induce long-lasting antibody have rigid surface repetitive structures spaced at 5 to 10 nanometers. It's an interesting comment. And they say the corona spikes are, embedded in a fluid membrane. They're far apart. And they say maybe this is a problem for all coronas because you get reinfection with the common cold coronas, every few years. The antibody response is not durable. And some have ascribed that to antigenic variation, but I don't know, it may be masking the lack of durability of the antibody response, right?
Does SARS-CoV-1, though, and the fact that they found long-lived responses in those patients kind of go against that?
Did they find B cells, long-lived B cells?
It's a good question. I'd have to look.
I know they found T cells.
I found T cells.
For 17 years later, they found T cells. I don't know about B cells.
Yeah,
That's
we
a
should
good
look
question.
and see.
Because that would be important for that argument. Yeah.
Yeah. So, I guess with a 5 to 10 nanometer, it's almost like they're saying it's about, the antigen and whether it can cross-link.
Yeah.
Yeah.
They say here that antibody responses to SARS-CoV-1 are also short-lived. So I don't know if anyone's looked in the bone marrow. Probably not, but they just look at antibodies. But what's I going to say? This would be a problem with HIV then because those spikes are far apart too.
Right.
If that's the argument. And of course.
That's interesting.
Anyway.
Yeah.
And they say the holy grail of vaccinology is the generation of LLPCs. I like that statement.
Yeah.
The holy grail.
Okay.
Well,
that's because, in their introduction, they talk about protective sterilizing immunity.
But sterilizing is at a mucosal surface. That is not in bone marrow.
I know.
Right?
I know.
I think that's a very high bar.
Exactly.
Yeah.
So I wonder, do we have data? Probably not. But what happens if you have infection with no vaccination? Do we generate these long-lived population? Is it generated to the similar extent? And is protective?
You probably don't have a huge number of those people who have also given bone marrow aspirate.
I think what would be neat is if the Elebedi group who published that, yes, SARS-CoV-2 infection does do this. They go back to those same people if they can recruit them, and they just look again and they see.
Yeah. Because I think that the one caveat, so they did a nice job in the discussion of this paper talking about some of the limitations. And one is, quote, "We cannot rule out the possibility where a subset of Pop B may be an intermediary population on the road to long-lived plasma cell maturation." And that, they're there, right? And they're categorized wrong based on the markers that they have. So what if the Elebedi study said, yes, they were there because they were using different markers or because they were looking more generally? Then they're both right, but they've just subdivided a population in such a way that it's not in this one bucket that they see these cells.
Exactly. Yep. And I think that goes back to, CD19 seems to be the marker they use to distinguish those populations. And what do we really, what do we know about CD19 and transitioning between those two? Yeah.
Anyway, it's provocative paper, right?
Really.
Oh, it's super interesting.
Super interesting. And
about.
It obviously suggests many other experiments that you should do with the idea of improving these long-lived memory cells.
I think you need to find out first, is that what we need to do? Right?
So what's the experiment you would do to do that?
Well, it would have to be a correlation between, it seems very difficult to do, but people who get really, I guess you could do a mouse experiment, right? You need more or fewer of these challenge
but then
and see if one of the populations is more protected than the other.
But what you have to hope is that you can you can say, this mouse population is the same as the population we see in humans.
I guess what you would do is you would take a normal mouse that's never been exposed to an antigen, and then you would populate the bone marrow with these purified cells that are either in this population or not, and then challenge and see whether having those around would be more protected than not having them around.
Right.
But certainly people have looked for long-lived B-cell memory in mice, vaccinated with SARS-CoV-2.
Yeah, I'm sure they have. I don't know the B-cell literature as well, but I know this is one of those places, at least in the T-cell literature, where the differences between mouse and human are immense in terms of markers.
Yeah.
Okay. Yeah.
Well, and if you could do it in a mouse, to Cindy's point, it would also be neat to do influenza, to do the tetanus vaccine, and just see if you see increases in those populations in the same way or not.
Yeah. Well, or to do the same antigen different ways, and see, is it the antigen or is it the delivery method? Right.
Because sustaining long-lived plasma cells relies on a lot of chemokines and cytokines, IL-6, TNF-alpha, IL-21. So, BAFF, APRIL, all of these things that matter. And so do those modalities have different levels of induction of that?
So, Cindy, you don't want to just do a study with protein-based spike?
Well, what would you do in that case? Just see whether just the Novavax generates these. Yeah. And then over time, just follow the patients who got, or the healthy people who got either type of vaccine and whether or not they got infected and what the outcome of the infection was.
Yeah, that's the idea. I think antibodies in those patients decline also. I don't remember the specific study, but I think it's been looked at and they go down just like the mRNA vaccines. But whether there's anything in the bone marrow, I don't know. I think that would have been interesting.
That would be complicated because then you don't have the T cell arm, right? So then we're comparing just the antibodies to antibodies and T cells.
Just look at long-lived antibodies creating cells in the bone marrow by the same methodology they use here.
Yeah.
Anyway, I thought this was cool.
That was great.
Very cool.
All right. That is Immune number 86. You can find show notes at microbe.tv/immune. If you have any questions or comments, you can send them to immune@microbe.tv. And if you enjoy these discussions, you can support them financially. We'd love to have your contributions. Microbe.tv/contribute. Cindy Leifer's at Cornell University, at Cindy Leifer on X. Thank you, Cindy.
Thank you. And I want to encourage people to write in with questions or comments based on the discussion that you heard.
Yeah.
So we don't get a lot of emails, but we would love to address the thoughts of the listeners.
Yeah. Steph Langle's at Case Western Reserve University, at Stephanie Langle on X. Thanks, Steph.
Thank you. This has been lovely.
And Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.